Bismillahirrahmanirrahim. We already discussed about insulin synthesis up to this process. This is the third video. Within the nucleus, replication and transcription occur, and after transcription, this mRNA synthesis occur, and this mRNA come out the nucleus through this pore in the cytoplasm of the beta cell of pancreas. This is the beta cell of pancreas. This is the cytoplasm. We know in the cytoplasm there are ribosomes. There are many organelle and ribosome also present. So mRNA associated with this ribosome causes synthesis of peptide or protein. This is protein. This mRNA enters into mRNA associated with ribosome. This translation occur and elongation of protein occur. So this one is elongated part, and elongation continues. This one is elongated part. This elongated part enters into the endoplasmic reticulum, we already know, and after properly elongated part of the mRNA, this protein part, the elongated protein part or peptide part, is properly folded within this endoplasmic reticulum. This is elongated part, it means this is the protein part or peptide part, and this is properly folded in the endoplasmic reticulum, and properly folding occur by this disulfide bond. This disulfide bond causes properly folding of the elongated protein. So here in the endoplasmic reticulum, here is initial segment, beta chain, C peptide, and alpha chain, and properly folding by this disulfide bond. Now in the endoplasmic reticulum, there are endopeptidase enzyme. This is the endopeptidase enzyme in the endoplasmic reticulum. Now this endopeptidase causes separation of the initial segment from this pre-proinsulin. We already know initial segment, beta chain, C peptide, alpha chain, and disulfide bond together called pre-proinsulin. Now this initial segment is removed by the help of endopeptidase enzyme in the endoplasmic reticulum. So now after removal of the initial segment, this part is called proinsulin. Pre-proinsulin initial segment with proinsulin is called pre-proinsulin. After removal of the initial segment, it is called proinsulin. Proinsulin means beta chain, C peptide, alpha chain, and disulfide bond. It is called proinsulin. And with initial segment, beta chain, C peptide, alpha chain, disulfide bond is called pre-proinsulin. So now after removal of the initial segment, we get this proinsulin structure. Now this proinsulin is enters into the folded structure of the membrane of the endoplasmic reticulum. This endoplasmic reticulum membrane is folded properly like vesicle occur, and this proinsulin enters into the vesicle or folding structure. This folding structure or vesicle fuses with the Golgi complex membrane, and this proinsulin enters into the Golgi complex. This proinsulin enters into the Golgi complex. Now in the Golgi complex there are also endopeptidase enzyme activities, and in the Golgi complex this endopeptidase causes separation of this C peptide. Suppose C peptide cutting here as well as cutting here. You just see follow here that C peptide is removed. This separation of the C peptide occurs in the Golgi complex by endopeptidase activities in the Golgi complex. So ultimately we can get this structure: active insulin, beta chain, alpha chain bind and with double disulfide bond. this active form of insulin also present in the Golgi complex. I did not show you here, but this active form of insulin also present in the Golgi complex. I just show you here pro-insulin, but C-peptide also removed here. In the Golgi complex, C-peptide also removed. This structure also present in the Golgi complex. This is the active form of insulin. Now you just follow that here also membrane of Golgi complex is folded, and active form of insulin or proinsulin present, and ultimately budding occur, and this folding structure separated in the cytoplasm. Here may be proinsulin present or active form of insulin present. If active form of insulin present, no separation or no other modification occur. But when proinsulin is present in the Golgi complex folded membrane folding, here endopeptidase activities occur, and this C-peptide removal occur in the vesicle. Suppose here proinsulin structure, here C-peptide present in this vesicle. Also endopeptidase activities present and separation of C-peptide. But when C-peptide removal within the Golgi complex occur, here no modification occur. But when this proinsulin present within the vesicle after separation from the Golgi complex, this vesicle separated from the Golgi complex. Suppose in the vesicle there is proinsulin, it means with C-peptide there also endopeptidase activities occur. But when separation of the C-peptide within this Golgi complex, this active form of insulin present in the Golgi complex, no endopeptidase activities occur here. I think you understand about synthesis process of insulin. This is the active form of insulin, and this is the secretory vesicle. This vesicle ultimately causes release of insulin, but release of insulin, action of insulin, it is the pharmacological process. You will understand later, but up to this you have to understand. You just know endopeptidase activities within this endoplasmic reticulum and cutting occur. Endopeptidase activities within this Golgi complex and cutting occur. Endopeptidase activities may also present in this vesicle, and may cutting occur when needed. Here in the endoplasmic reticulum, initial segment is removed. In the Golgi complex, C-peptide is removed. If C-peptide is present in the vesicle, secretory vesicle, if C-peptide present, here also endopeptidase activities occur and C-peptide is removed. So active form of insulin and C-peptide ratio is equal 1:1. If one active form of insulin present, one C-peptide is present. That's why we can count C-peptide, and we can count active form of insulin, number of active form of insulin within our body. Thank you.